Hello and welcome to Tuesday Talks at Merryspring Nature Center for 2026. My name is John Fromer, the Program Director and your host. Today's Tuesday Talk is generously sponsored by Camden River House. Recordings are made possible by the Cascade Foundation. You can find today's talk and previous talks on our YouTube channel. Merryspring is open every day of the year from dawn to dusk free of charge. Find out more at merryspring.org. Talk was presented on Tuesday, March 17th by Bob Gastaldo on the non-analog forests of the coal-forming Ice Age.
We're good. It is afternoon, so good afternoon. What, lights?
I'll do lights.
Camera, action. You got the lights? But I get the light in front of me, right? Good afternoon, everybody. Glad you could make it. I'm glad I'm be back here. What John asked me to do is to talk a little more about ancient forests, because we are Northern Hemisphere chauvinists to begin with, because this is what we consider to be a normal forest. And we also have a very short amount of perspective on time and the kind of forests that have dominated the planet since their inception in the middle Paleozoic. So the image that you see on the screen is something that I put into Gemini, Gemini being an AI program. And I said to Gemini, construct me a forest that has the modern equivalents of ancient trees that we'll be talking about today. The things that we find in the forest is what we'll see in Maine, lycopodiums, ground lycopods, and horsetails that the ravage of our lawns when they get established, and ferns and the like. And this is what they came up with. And when I wrote the prop, I put in quite a number of modern taxa, Equisetum hyemale, Lycopodium clavatum, and the thing that first came out from the prompt was, oh, you want me to make a Carboniferous forest? And I went, yeah, that's essentially what I'm asking you to do. And this is what came out of it. And we'll be talking about, it looks real, right? It looks like it could be anywhere. But this is what we're going to be talking about today, this non-analog forest of the coal-forming periods, because the trees that existed then and the ecosystems that are there are very, very different from what we think about the modern world. But as come on, oh, back. We got a lag, right? We got this lag. Come on, back. Right. What I like to do back, good boy. No, this is the transfer of data between you and I. I'll just hit the button. What I'd like to do is to give you a perspective of where we're going, because deep geological time is difficult to fathom at best. Now, 4.5 billion years ago is when we have the first record of rocks and age dates and minerals on Earth. And that's way back here in the Precambrian. We are here, right at the end of the geological time period. And we're going to be going back to here, the Pennsylvanian period, or the interval of time when it was the last major coal-forming period under glacial conditions. So the period of the Pennsylvanian is the same length of time as it is over the last 24 million years from the late Oligocene to the present, when Antarctica started increasing its ice cap, that we've gone through glacial and interglacial times, and also coal-forming periods throughout the Miocene and late Oligocene. So the same amount of time that we have seen the last glacial and peat-forming conditions happened 320 million years ago. Right? So we've had almost 300 million years of time between the last major coal-forming period and major glaciation and what we're going through now. And if we look at how we've reconstructed those glacial times, we see that similar to what we have in the Miocene, Pliocene, Pleistocene, and recent, we have cold, coal-forming times, glaciation, and then warm times, and cold times and warm times. And we fluctuated back and forth from major glacial periods to a warming trend, to glacial periods and warming trends. The same kind of oscillation that we see in the last 24 million years. But again, millions of years are difficult to comprehend for most people. So, as I've done in previous talks, I like to think about these as if we were to take a 1,000-sheet toilet paper roll and assign 4.25 million years per sheet of paper, in 1,000 sheets of toilet paper, we could go back to the beginning of time. Where we are on that toilet paper, and I know you're never going to look at the toilet paper roll again similarly, right? You're always going to think, where am I? And we'll get there in a minute. If we look at the toilet paper roll, we, right, today are just a few fibers at the end. If you take your fingers and kind of go and pull off maybe 4 or 5 fibers, that's us in geological history. Again, very difficult to comprehend. Our species, Homo sapien, is less than 0.06% of a toilet paper sheet. The first use of fire is 1/10 of a toilet paper sheet. So you can't even get a good fire started with 1/10 of a toilet paper sheet. The Quaternary, which is 2.5 million years ago, is only half a sheet of toilet paper. And then the Antarctic ice sheet that we just talked about, which is this last glacial period, is only 2.2 sheets of toilet paper. Right, we've got 998 pieces of toilet paper to go to get back to the beginning of time. We're going to go 68.3 sheets of toilet paper back in time. So next time you put a new roll in the toilet, you know, you can count on the wall or keep track on the wall, 5, 10, 15, 20, 25. Oh, I got to 68.3. Good luck with the 3.3. And this is where we're going to go today, to these different kind of forests in the late Paleozoic, which are dominated by spore-bearing plants. Not by seed-bearing plants, but by spore-bearing plants. We're going to go to the middle Pennsylvanian, and primarily for today, but we'll talk about the plants that make up all of these coal-forming environments. Next. That's like a 4-second lag. I'll remember that. The major plants, the major trees, are the same group of plants that we see today as ground cover. Lycopodiums, club mosses. Club mosses, right, you'll find as ground cover in Maine woods. The largest club mosses that you'll find are Southeast Asia. They get to be about 6 feet or so tall. In the Carboniferous, these trees got to be 21 meters to 40 meters tall, with diameters of 1 meter or more. So we're talking, 40 meters is almost 150 feet in height, taller than any tree that you'll see out here. This lineage established late in the Devonian, which again is much older than the Carboniferous, and there are 3 major lineages that show up. There are the Lycopodiales, which we see today in Lycopodium. These are some of the major trees. The Selaginellales, which is another group of small little climbing or vining lycopods, Selaginella, and Isoetes, which have this little cormose base at the bottom, put up several leaves at a time, kind of look like a little onion in sprouting. but they were completely spore-bearing and not angiosperm in origin. The isomotes evolved into these upright and tree habit forms. This image is from Joggins, a UNESCO World Heritage Site, of one of these trees preserved erect and surrounded by sediment with its rooting structure called stigmarian axes here. And this tree has a bipolar personality. And I say that because it's true. Right? If you look at the anatomy of the branches and the leaves, and you look at the anatomy of the stigmarian root and its rootlets, they're exactly the same. There's no real differentiation between internal anatomy. So you can think about this plant as having branches and leaves at the top and branches and leaves in the bottom, and this stem between the two. Very, very odd construction. And these plants all produced by spores, so what they needed to continue propagation are wet habits. Like the ferns we have today are lycopodium today, the spores are produced, subdivide, and then the alternation of generations between the sporophyte and gametophyte generation occur. The same thing with these trees, which of course trees today don't do. These were the giants of the coal fields. There are many different growth forms. The two principal growth forms are either a determinate growth, where the juvenile tree increased in width, grew up until maturity, then produced a crown of leaves and branches, at the end of which were large cones. And those cones are what produced the spores. There are the indeterminate growth groups, similar to this growth habit, and that means that the plant grew, produced lateral branches, continued to grow, and it did not come to a finite end until something happened or it just got too old like me and decided it was going to become part of the forest floor litter, right? The taxa exhibit either this development of determinate or indeterminate and these varying growth architectures from dichotomously branched, multiple dichotomously branched crown to things in a genus called Sigillaria that branched one and it produced a tuft of leaves, long leaves, and the leaves could be up to 1 meter long. And the juveniles and the mature plants, they are about a centimeter or so, and so it changes its growth habit. But this is the principal plant that accumulated into our economic holes, because the biomass is enormous. What's unusual about this, and we'll see this later, is that you would think a tree that would be the diameter of the table in front of you would have a lot of wood. It doesn't. The amount of wood is probably a couple of centimeters, maybe 10 centimeters in diameter, and the rest of the tree is all soft tissues, all parenchyma and some lignified periderm or bark tissues. Very unusual set of characteristics. The horsetails or sphenophytes. Today we think about Equisetum arvense. This is probably the most common one in our forests. It has a jointed or segmented stem with whorls of leaves or branches that develop around it, and the same growth habit occurs in the Carboniferous. Here are some early Pennsylvanian horsetails that have their leaves at the nodes. And in the case of the late Pennsylvanian and Permian, these plants could attain a diameter of 1/2 meter or more, and they were woody. You don't think about horsetails as being woody, but the fossil examples have an immense amount of wood. There are two types of growth habits. Things such as the sphenophyllales, which are here, some of the South African work we published on, are ground cover and climbing vines, and they have very thin stems around which the leaves or cones form. And then we have the tree examples. This is a reconstruction of a tree from the Chemnitz, a volcanic fossil forest in Germany. based upon the diameter of the trees and actual trees blown down in this volcanic eruption, reconstructed, at 70 feet tall. That's pretty impressive for something that is our ground cover today. The unusual thing about horsetails is that they are clonal for the most part. We have examples here in Chemnitz of individuals with rooting structures, but most other horsetails are clonal, meaning that they put out an underground rhizome or stem, right, that goes, and then they put an aerial stem up, and then it continues on and puts an aerial stem. That's why you can't get rid of them in your lawn or garden, because they just continue to develop this underground stem. It's like whack-a-mole. Oh, I'll just cut that one off. And the ecosystem goes, good, I'm going to go over here. Right? So it's difficult to do. The thing that is unique in its architectural of the horsetails is that all of the structures are whorled around the nodes. There's no spiral arrangement to leaves. It's strictly whorled. And the whorls can be as beautiful as this annularia, a lateral branch, the secondaries, and beautiful leaves, a pith cast of calamites, which is the main stem of this plant, showing again this node, internode faction. And these plants do not make up a large percentage of the peat forming or peat accumulating ecosystems in the coals. They are found mostly outside of the peat environment in mostly clastic, but I mean by mineral soils. At the same time, boom, there we go, the ferns are around, and we have many different fern groups. These range from ground cover, like the Botryoidales and a number of other families that are extinct, to the same families that exist today in Hawaii and Southeast Asia, the Meradiales, which this is a Meradialian tree fern, and this is a Blechnum from Europe. And the thing that distinguishes these tree ferns is that they are understory plants. They grew similar in size to what we have today, maybe 10 meters at the most, and take a sub-canopy position within the forest. Now, ferns develop either by a single spore, heterospore, or homospore, where the single spore type produces the gametophyte, which then produces the sporophyte, or heterospore, where they produce a male and female set of spores, which each then produce their own plant, which then produces eggs and sperm, which are fertilized to get the sporophyte. And we have examples of both of those life strategies in the Carboniferous forests. Now there's a group of big sensu lato group, big umbrella group, called the seed ferns, or pteridophytes, which are actually gymnosperms. There are two main lineages in the Carboniferous, something called the Lygenopteridales, the Lygenopterid ferns, and the Medulosans. Lygenopterids here give you an idea of what the central anatomical details are. They have secondary wood around a central pith. They also have these very strong bands of resistant sclerenchymatous tissues that help support it, because most of these were ground cover or lianas, or vines. And having that additional support around the edge of the axis allowed them to climb. Sorry, should turn that off. It's probably a scam, right? Lygenopterids are unique in having the bands showing up along the axis, as well as a number of different very small pinnate leaf architectures. They reproduce by a pollen organ, something called prosoptheca, where in this case, you can see that there's a head, and all of these little things are pollen sacs. And the pollen sacs are very different, the pollen produced here, as we'll see, very different than what it looks like to be a fern spore grain. And then they produce a very unique seed called this in the case of Laginostoma, that has a series of extensions around the integument and this laginostome, or little chamber where the pollen would come in, then germinate and fertilize the egg. Very different. We don't have any of that today. The second group, which is more of the major group, are called the Megillosins. These are woody trees that, again, could be up to 1/2 meter in diameter. But the wood in these trees is unique because when we think about a tree with wood, we think about a central part of the tree, and the wood grows around that tree, right around that center, in circles. These have multiple vascular strands, each of which produce their own set of wood. So you can almost think of this as an aggregation of individual stems within the big stem, producing their own wood and producing their own laterals. These are reconstructed with adventitious roots, which we have in the fossil record, so that they needed something to help them support this unique architecture. They got to be shrubs and part of the understory, and they range wide ecologically from the coal swamps, from peat mires, out into more well-drained soil conditions. They're called seed ferns because the leaves look like ferns. And when they were first discovered in the 1800s, it was, oh, this is a fern. And then in about 1890, Scott found a seed attached to it and went, uh-oh, they're not ferns. What could we call them? They have seeds, right? A equals B, B equals C, we got seed ferns. Which is a misnomer because they're actually gymnosperms because they reproduce by these seeds. They produce pollen organs that are very different than the Laginopterans. You can think of these like they're campanulate-shaped bell structures with all of the pollen sacs hanging down and releasing the pollen out from them. A colleague in Pennsylvania ran some simulations a number of years ago and found that the water conduction in the Megillosins, based upon the anatomy, the size of the water conducting cells and the like, had the same function as modern angiosperms. So these things could pump water through them significantly, which tells us that the angiosperms did nothing new, they just evolved and used something that had already been in the genetic code of the plant kingdom. So when we look at these areas, we don't always find fossil leaves. We don't find fossil stems. About 5% of the rocks actually have megafossils in them, which is not a whole lot of area. Yes, we know these ecosystems better because they're associated with economic coals. So there's been a lot of economic coal development since the Industrial Revolution, so there's a lot of fossil plants of Carboniferous age that are known. But we know them more from their reproductive structures, these pollen and spores. And just to give you an idea of what the pollen and spore look like, these are two different spores from the club mosses. And you'll notice that they have a trilete mark to them. Most spores have trilete marks because when meiosis occurs, the one mother cell produces 4 spores and they're all attached together. That leaf mark tells us where the other 3 spores were. When they fall apart, these become the weak areas for germination to come through. So the trilete mark is very characteristic of spores. These lycophytes have a very punctuate or very ornamented surface to them and have this dense outer ring around them that could have helped them disperse by wind. That's a hypothesis. The horsetails, on the other hand, are like vanilla. They are the vanilla of the kingdom. They have no sculpture to them. They're very thin-walled. They have a little bit of sculpture, but they're much larger than the lycopsid ones. And these are thought to just have been dispersed down to the ground without any ability to have been transported by wind. Tree ferns and ferns, again, have a nice trilete mark, and there are hundreds and hundreds and hundreds of varieties of these kind of punctatus varieties of Lophotriletes and the like. They go in and out of the fossil record, which we'll see in a minute. They go in and out of the fossil record as climate changes. As the edaphic conditions, precipitation, and temperature change, we can either get a flush of tree fern ecosystems or a lycopsid ecosystems. But the thing to remember is that all of these plants coexisted in the same forests at different structural levels, and we'll see that in a minute. The gymnosperms have a wide variety of pollen grains, because remember, these are now seed-bearing plants, they produce pollen. And we can see the difference between things such as the medullosin pollen and Florinites, which is a pollen grain of a conifer relative. And you'll notice, okay, when are our cars going to become yellow again? Right? Like a couple of weeks. Once the pine comes out and produces that pine pollen, which are saccate, they have two little Mickey Mouse ears or air bubbles on the outside of the grain, they get windblown, they settle on our cars, and we curse. At least for a couple, I curse. I don't know about you. Gotta get rid of that stuff. Well, the early conifers and conifer relatives also have this same mechanism. This is an air bladder around the central cord of a chordites, which is a pre-conifer plant that we'll talk about in a second, which also coexists. These conifers are woody. They have the same anatomy, the same vascular structure as you would see out here. John was talking about before about these pine trees being felled years ago. They have the same anatomy. It would be very difficult to distinguish sections of chordites from modern pines, because once that architectural growth habit of wood production sets itself in the evolutionary history of the group, it is successful, it stays there. Right? Don't fix something that ain't broke. Right? And the conifers have not fixed something that ain't broke. These trees could get to be anywhere up to 30 meters or more in height. And we have examples, particularly up in Canada, where we have trees that are 25 meters long in ancient river channels that were log jams. They produce, though, rather than the little leaves that we see on conifers, they produce large strap-like leaves off the branches. And they produce these very elongated cones. Now, when you think about a conifer, you want to think about a nice compact cone. In the paleobotanical record, those don't show up for another 150 million years. Most of the cones are these central axis with the lateral bracts and seed-bearing structures or pollen sacs on them. A couple of weeks, we're going to see some of the birch trees come out with their long, elongated catkins. You can think of this as similar to a catkin in appearance. Obviously not in structure, but in appearance. It's something that probably hung down and caught pollen. And chordites have a place in the swamps, but they're also known mostly outside of the swamps in better drained mineral soils. Now, one of the last groups, and most of those are all northern hemisphere, one of the last groups, which is strictly a southern hemisphere gymnosperm, are the glossopterids. And you may recall from when we actually had science classes that continental drift was first thought about by Wegener because Glossopteris, this plant, occurred in the fossil record of South America, Africa, Antarctica, India, and Australia. And he couldn't rectify how the same plant could be distributed over all of these continents unless those continents had once been together. This is the culprit right here, Glossopteris. They too are a woody tree. some of the material that we have in South Africa, 1/2 meter in diameter, large trees. The male and the female born on separate plants, and these are Glossopteris leaves with their reproductive structures, which are very unique and odd, multitude of odd. I mean, if you're the same genus separated over large geographical spaces, the evolutionary processes are going to give you multiple versions of the same successful strategy. So Glossopteris is a female Glossopteris. The different families are separated based upon their reproductive organs. But we're not going to talk about those because we don't live in the South. But we are going to talk about its reproductive history because it gives us a very good idea about how these plants operated similar and dissimilarly to what we have today. So in the case of Glossopteris, we have a large tree that produces a central dictyostele with secondary wood on it. Those trees were affected, similar to trees today, by insects. This is a cavity that has mite frass on the inside and wood rot. Those leaves then fell onto the leaf litter, fertilizing the soil. They produce pollen and seeds. In the case of the rest of the plant, we have invertebrate herbivory on it. Epiphytes and bryophytes grew on there. We have evidence that this was a microcosm in and of itself. And in the summertime, all that occurred on the plant. We have evidence in the fossil record that during the wintertime, all those leaves dropped off. We have lake deposits that have annual accumulations of leaves, just like you would think you would see in a lake here in Maine, over and over, right? The leaf drop going to the bottom and being set in the So what we are able to do is once we have a large collection of fossil plant material of multiple character, anatomy, morphology, ecology, reproduction, we can, and a geographical understanding of their distribution, we can put these forests back together. And that's what I'm going to want to do for you today, is put these forests back together, just to give you an idea how bizarre, unusual they are. But they were successful for 30 million years. So, could that be that bizarre? This is some work we published on too many years ago from the Black Warrior Basin in Alabama, and we're going to go down here at about 320 million years ago to something called the Westphalian A, where we were working the Black Warrior Basin in the Mary Lee coal seam, the largest economic productive seam in Alabama. And we were fortunate enough to work with the Drummond Brothers Coal Company, where they would allow us on to the top of the coal as they stripped the rock off the top of it, because the forest floor litter and standing trees were preserved above that coal. So I had two students go back every couple of weeks as they moved the drag lines along and survey quadrats, 10 meter by 10 meter quadrats, to get an idea of what the composition of the floor was. And using some statistical analyses, we found that there are individual locations that are dominated by canopy cover, which are mostly lycopses. Locations that are dominated by understory, less than 50% canopy. Ground cover dominated locations, less than 60% canopy locations, similar to what you'd expect in a modern forest. This is a heterogeneous, it's not homogeneous, it's not Irving, right? Irving's wood forest or Weyerhaeuser's forest. They're not lined up individually, they don't show a pattern, and they're not all the same plant. But because of the changes in edaphic conditions and soil conditions and topography or drainage, we have groups of plants occupying different spaces in the forest. And this plot here, which is a half a kilometer in this direction and 2.5 kilometers in this direction, give you an idea that we had an area that was dominated by canopy, surrounded by areas that had less canopy, or just strictly ground cover. We don't think about forests where you walk out and all of a sudden get to a meadow. Yeah, sometimes you get to a fern meadow. But you don't think about fern meadows as being co-equal in a forest structure. And I have to remind you that these are all peat accumulating environments similar to the peat forests that you would find in Southeast Asia. So this reconstruction that my colleague Steve Greb did at the Kentucky Geological Survey for us and his co-author kind of gives you this idea that the forest is really open. It's not a closed canopy forest. And that the major dominant trees are almost like an old growth forest, where they're big and then there's a lot of space between them. So this is Westphalian A. My colleague at the Smithsonian, Bill DeMichael, worked primarily in the Westphalian D at about 307 million years, so about 13 million years younger. 13 million years plus or minus a day. Who cares? And he worked underground coal mines. Various coal companies allowed him access to the Heron No. 6 coal, which again in Illinois was the largest producing economic coal for the area. And underground, he and the geologists at the Illinois Geological Survey would walk the adits. Now, I don't know if you're familiar with the term adit, but if you go down into a mine, the idea is a pillar and room mining structure. So this thing had been complete. I'm going to put you in the middle of a coal right now. We're all in a coal, but right now we're going to mine the coal. We're going to mine out this room of coal and leave the wall or an adit to support the roof. Then we're going to go and mine the next room and then leave an adit to support the roof. So there's all these tunnels from room to room to room supported by coal underneath it. So they're able to go into these rooms and evaluate the fossil flora on the ceiling and map those for over, I can't remember how many square miles of mapping they've done. But Bill and colleagues published a paper a number of years ago where they were able to show adjacent to a river channel that had cut into that coal at a later time, the Walshville Channel. That adjacent to that channel, there were these various, again, groups of plants, ecosystems, that were dominated either by very tall growing seed ferns, similar to the height of nipa palm forests. And I don't know if anybody's familiar with nipa palm in Southeast Asia. That plant produces a small little cormose stem, and then the leaves grow to 7 meters in height. I'll show you pictures from our work in Southeast Asia. So this area is similar to a nipa palm swamp, right? Adjacent to it, though, are locations that are dominated by tree ferns. And if we look at the distribution of these ecosystems, we can go from tree ferns and lycophytes in pink to strictly lycophytes to pink to lycophytes to these Neuropterus flexuosa, these nipa palm-like plants. And you'll notice that similar to any other forest, it's heterogeneous, but different than what we saw in the Westphalian A in Alabama, different composition. We've been working recently in New Brunswick, Canada, in the middle Pennsylvanian and Westphalian Sea, which is older than Illinois Basin, younger than Black Warrior Basin, along the Bay de Chaleur, up in New Brunswick, where a group of rocks called the Clifton Formation is exposed and continually gets re-exposed as the cliff face falls down, dangerous to be there, and it pushes this back. It's a beautiful place. when it's not windy or raining or cold. We did field work last August and we had frost, so it was a little chilly. But you'll notice that if we look at this drone image, you see that there are large sandstone bodies, and I've just highlighted in little red circles standing trees. We have forests preserved in these rocks of standing trees, but not just standing trees. We have at least 7 different horizons, 7 different levels of forest development with only 16 meters. So 40 feet of rock have 16 forests that are preserved, one of which we can trace all along the outcrop, for a couple of kilometers, about 2 kilometers. And these plants, unlike the Alabama plants and unlike the Illinois plants, did not grow in a coastal community. There's no evidence of tidal activity. There's no evidence in geochemistry of estuary waters or marine waters. This is a strictly freshwater, inside the continent forest. Okay? Very different. And you'll notice that right about this position, right beneath the forest that we'll talk about today, we have a volcanic ash horizon. And we have zircon from that volcanic ash, which means we can get a uranium-lead radiometric date for exactly when this forest grew. Pretty amazing, because who knew, right? When we were there last August and came across this, as I taught my students in South Africa, what doesn't belong here? And when you run across a centimeter white-gray band that is muddy clay, well, that doesn't belong here. Everything else are reddish siltstones or greenish siltstones or sandstones, like, no, that's volcanic ash. So those analyses are being done right now in the Jack Satterley lab in Toronto with my colleague Sandra Kimo. The forest has a unique burial history. This is a 3-meter tree here, right there, with its rooting structure right above the devitrified claystone. And you'll see that it is enveloped by mudstone and then enveloped by sandstone. And this tree is 0.65 meters in diameter. Now, if we use the equations that Carl Niklaus at Cornell developed in the 1990s to estimate the height of a tree, a non-woody tree, this tree comes out to have been 21 meters tall. Yeah, a big one. And we have trees here that are almost a meter in diameter also. Extremely tall trees, but only 3 meters of the base are preserved. And we call a trunk with its base a bole, B-O-L-E. And you'll see in this case that the bole was surrounded by sediment in at least 6 different phases of sedimentation. And at one point, along the top here, we have well-preserved fossil leaves, telling us that the trees were alive when they were first buried and died, that all the aerial parts then fell off at this level, were preserved, and that we continued to fill around these trees for up to 3 meters, it's actually more because that's not decompacted. Sediment has water in it. When they become rock, they get compressed and the water goes out, so more than 3 meters. And the inside of that tree then is filled with sediment. So it preserves it in this 3-dimensional structure as an internal cast. So just to give you an idea, we have a tree that's nicely, happily growing. All of a sudden, its rooting system in lower base is covered in mud, which then kills the tree. We know that you can't smother rooting systems and expect the tree to live. We lose the canopy vegetation, and then for some reason, which we can talk about later, accommodation or increased space allows for sediment to build up around that tree to a height of more than 3 meters. We have, I said, we have beautiful fronds, beautiful tree fern fronds, seed fern fronds. These are the lycopod branches. This is a frond of a liana, seed ferns. Beautiful, beautiful stuff where it's preserved. It's not preserved everywhere, but where it's preserved, it's exquisite, which tells us that they died, they didn't have a whole lot of time to decay, and were buried and taken out of the system. But that's not all we have. We have skeletons of at least 48 tetrapods, ranging in size from 10 centimeters to 1 meter, preserved inside the bottom of these trees. Here you can see a postcranial skeleton of a tetrapod. There's the spine, those are the legs and the like. We also know that the inside of the trees dried out and got wet, and dried out and got wet, and dried out and got wet, because at this particular level, we have raindrop imprints on the sediment. We also have evidence of annelid activity, soft-bodied worms with all of these trace fossils. Huh? How does this happen? We could go into a long discussion of how it happens. It happened. We'll just leave it there for the moment. We can talk about that later. But in essence, once the tree was surrounded by sediment and the top of the tree fell, the inside, all those soft tissues decayed, leaving this hollow void into which animals could fall or were drowned. Because in order to fill the inside of that tree with sediment, you had to have floodwaters come across the surface, fill that tube, the sediment settled out to bury whatever was in there, and then dried out, and then weathered and dried out. We have most of our animals within the lower meter of the trees, and tree to tree differs, right? Not every tree is the same. It's a very complex history of how these things were preserved. So let's take a look at one tree. This is a small tree, a 2-meter tree, where there's a basal 25 centimeters of sediment, where there are 3 levels at which vertebrate remains occur. There's one level where those trace fossils occur. Another level, higher up, where more vertebrates occur. And the amalgamation of ecosystem diversity ranges from little microsaurs. This is a CAT scan of a microsaur that is, the skull is 1.5 centimeters, beautifully preserved, to synapsids that are a decimeter in size. Now you say synapsids. What are synapsids? Because that's a term that most likely is not often used. We know reptiles. We know mammals. Synapsids are the evolutionary group between those 2 clades. Synapsids have characteristics of reptiles, skeletal characteristics. They have characteristics of mammals. They have well-defined dentition. Reptiles, as you can see here, have one style of tooth. Synapsids have molars and incisors. Synapsids also have a rib cage like we do. Reptiles don't. Rib cage tells us that they had a lung capacity with a diaphragm that allowed them to breathe differently than toads and other amphibians. We have isopods. Everybody know what isopods are? Roly-polies. Roly-polies are preserved here. That's millimeters. That's 5 millimeters of a roly-poly. We have arthroplurids, which are millipedes. In this case, we have small arthroplurids. We don't have a very good image yet of the arthroplurids, so I put this up there. But arthroplurids in the Carboniferous grew to the length of this room, with a diameter half the size of that table. Again, who knew, right? We have amphibians, something called befeated, which are extinct, the skulls of which are watermelon-sized. The postcrania makes this animal, and we have it all attached, 1 meter in length, right? The size of that table, in the bottom of the trees. We have hexapods. Hexapods, diplurids. These are little blind soil animals that live in damp, dark places, well preserved on top of the bed. And then we got insects with compound eyes. Right? A treasure trove of diversity of a single forest at a single point in time. So what can we learn from only the inside of a tree? We've got herbivores and omnivores that are interacting with decomposers, which are interacting with scavengers, which are interacting with aquatic, semi-aquatic, and secondary consumers. Oh, I forgot to mention, we also have a freshwater shark in the bottom of the tree. Who knew, right? We have various tetrapods, primitive amphibians, labyrinthid amphibians, which are holdover from the Devonian time. Those are big predators. We have apex predators in there. We've got reptiles, true reptiles. We have synapsids. We have temnospondyls. All of these hierarchical structures inside. We've excavated 3 trees to date, including primary consumers, secondary consumers, and the like. So this is a really unique and exciting deposit. And we've only scratched the surface of it. We've got a couple of papers that are out for review at the moment, which may end up again in one of the national media because it's so exciting. So what can we conclude? If we look through the Mississippi and Pennsylvania into the Permian, about 100 million years worth of time, and we were looking at the Pennsylvanian, most of the major groups that be the dominant trees and plants in the Carboniferous forests evolved earlier in the Devonian and still live today. Except we don't have any lycopod trees. We don't have any horsetail trees. We don't have any cordite trees. We have tree ferns, they're very similar, but we don't have the dominant groups of plants 300 million years ago being dominant today, once the angiosperms evolved. We have numerous lineages that are extinct. Not surprised there. And right here at about the mid to late Pennsylvanian, we underwent an extreme warming event that changed the composition of the flora for another talk at another point in time. But that significant change moved us from a ice house climate into a hothouse climate. The planet went back to a ice house climate in the early Permian and finally came out at the end of the Permian into really hot times. The spore-bearing groups are the ones that dominated the forests. We don't want to think about the spore-bearing groups as the dominant plants in forests today. but they sure were successful for a long period of time. We also have the earliest examples in these forests in and out of peat swamps, of ginkgos, of early conifers, cycads. We now have evidence of cycads in these forests. And these are mostly found on substrates that were mineral soils and not in the peat. And last but not least, they come to dominance in the early Permian. The gymnosperms come to dominance in the early Permian after this warm period into the second phase of major glaciation across the globe. And it's that changeover to the gymnosperms, the seed-bearing plants, that are the precedent to the domination by seed plants up to today. So I'd just like to acknowledge all my colleagues who have been helpful in this. Matt Stimson and Bailey are at the New Brunswick Museum of Natural History, and they're the two that have been finding most of the material. Brian Hebert ran the drones for us. He works at the UNESCO World Heritage Site up at Joggins. Hilary Madden is a vertebrate paleontologist up at Carleton College, who has been doing all the scans with her students. Michael Regal and Paige Quinton are sedimentologists, geologists. Actually, Paige is an isotope geochemist, and they've worked on the sections with us. Duncan McLean and Court Evil are both palynologists. Court also does coal petrology, so we understand what the composition of the coal was and how it was formed. And then Lucy Smith works in the zoology, and she's been helping us with the invertebrate animals. So if it wasn't for these peoples, and a whole lot more, I couldn't bring you what we talked about today. So I'd be happy to take any questions.
Bob, this may be the entry to a, stay over there.
Yeah.
This may be the entry to a different talk, okay? But if you could transport yourself 300 million years into
I
the
wish.
future, what would you expect to be the dominant characteristics of anything found in the coal formation from today?
From today.
Yeah.
As I say, the lycopods are just the most biomass that you have in the swamp. Absolutely the most. So, because there are determinate and indeterminate growth forms, I'd expect to see trees the size of sequoias, or, so I've worked in Southeast Asia. I've worked in Borneo for a couple of field seasons. I've worked in the peat swamps of Sarawak in the early 90s as analogs for coal forming period ecosystems. And I would expect to find plants similar to what had been cut down by the early 90s. So, by the early 90s. So we never slept on shore. We had a boat. We slept in the middle of the river away from the mosquitoes, even though we had netting and the like. Didn't want to get malaria. And one night, beautiful sunset to the west, and we're on the boat, and I say to the person that we're working with at the forestry department, I go, it's a beautiful sunset, just look at the landscape. And you can see emergent trees, big tall trees above the regular canopy. And he goes, well, that's not the real forest, because he'd been working there since the 60s. And he goes, the real forest were the emergents that were another 50 to 70 feet taller that had already been cut out of those forests. So this is the kind of thing that I imagine the Carboniferous would look like, these enormous emergent trees with at least one or two layers of understory in this patchwork of groups of plants. And I think that's what we have here at Clifton, are these emergent trees, because they're so big and the periderm or the bark tissue is so resistant that once it hollows and it's confined by the sediment, they're just this well. Just come on, fall in, blah, blah. It's like a cistern or a septic tank, right? Well, it's got to be a septic tank because those things are preserved well, right? You got to get rid of the oxygen, you got to deter bacterial degradation. And they're only in the bottom, about the first meter. again, depending upon the trees. And the bizarre thing, and I know I could go on forever, the bizarre thing is that at the top of the trees, the uppermost meter of sediment has a soil formed in it. But not a soil that you would think of. The soil has limestone nodules called concretions, pedogenic carbonate concretions, that you would find in a more seasonally variable climate. So calcium and magnesium, pore waters were coming in, it was getting evaporated, they were concentrating in what's called calcic soils. It's like, what? There are no calcic soils next to them. But only within the tree do we have evidence for this heating, drying, rapid change in internal chemistry. It's bizarre.
What was the oxygen content in that time period?
So estimates on oxygen that have been done by Isabel Montanez and the groups are similar to oxygen contents today. So the talk that I gave a year ago, 2 years ago on fire, we talked about that in order for fire to happen and propagate, you need 16% oxygen in the atmosphere. Anything lower than that, it just kind of, it doesn't ignite and propagate. So at least 22% is what's been estimated, maybe 24%. And there are estimates that were higher because if you've got these invertebrates, like Arthropleura, that are the width of this table and the size of this room, the amount of oxygen they need to get into them, because they don't have lungs, has to be at a much higher concentration to cross that threshold and make them functional. So it might have been 28, it might have been higher.
Yeah.
But you can't get too high, otherwise, right, oxygen oxidizes, the tissues break down, right? That's why you don't want to get too much oxygen or high oxygen concentration in the hospital because boom, right? Other than that, yeah.
Wonderful. Bob, thank you. Any other questions?
Any measurement of CO2 at the time?
Turn that off.
Thank you for joining this Tuesday talk. For more information, please visit Merryspring at www.merryspring.org and also on our YouTube channel.